Audible, right? Hello. Right. So today we discuss the transgenic plants. Online. The transgenic plants. Now, transgenic plants means here there are the genome modified plants. Means here what are the characters? With the the transgenic plants are produced. Means here the transgenic plant. Correct. Right. Modified using the different tools of genetic engineering to insert the trait of our interest. Trait of our interest means who want to the desirable character, good plant, tolerant plant. What are the desirable characters we want to choose that one? These traits do not naturally occur, so we need to modify the plant's genetic material. An example of the first transgenic plant is the tobacco in 1983. So first one is the tobacco is a modified plant, and the transgenic plants are created for the numerous benefits such as the resistant biotic abiotic process to increase the nutritional value, another disease resistance plants, pathogen free plants. We have to require, we have to create these transgenic plants. See here, I shown to one picture about the transgenic plants. See here. So, so what are the transient plants? Means the transient plants, those are have their genetic makeup altered by the introducing the genes from different organisms like vectors. This isolation, the genes of insert from another organism such as bacteria or other plants, making any required modification and then introducing the altered gene into the target plant's genome to which is specific traits. So see here in this diagram, in this picture here, this is the TI plasmid with itself of the TI plasmid. We have to cut some enzymes, then enter into the another plant to modify the genes. So different methods to create the transgenic plant. There are various methods through which can be transferred the gene to the plant's genetic material. That is the agrobacterium mediated transfer. The agrobacterium, TI plasmid of agrobacterium tumefaciens is used to insert the gene of interest in the selected plant genome. The TI plasmid has capability to form tumor known as the crown gall disease. That is they call as a crown gall. The another method is the biologistic method, biologistic transformation. So in this technique, gold coated microscopic particle are used which carry gene of interest. These particles are released using the gene gun and directly enter into the cell nucleus with high speed. With help of that speed gun only, here the genetic material is transmitted to restricted genes. Another method is the electrophoration. This electrophoration is the electrical current used which create the temporary pores in the plant cell wall using genetic material. Another technique is the micro injections. This is very complicated technique of the micro injections in the transgenic plant to create the transgenic plant, which allow the directly insertion of foreign DNA into the plant cell nucleus. So means directly with help of the injections means micro injections, the required gene material is directly shifted to the target gene or target plant. Another method is the virus mediated gene transfer. The plant pathogenic virus is engineering in such way to that carry the gene of interest, and when the virus infected the plant, that the gene of interest get expressed into plants. So polyethylene glycol transformation that we call as the PG polyethylene glycol. So PG is the mechanical which is the applied on the plant that result in description of cell wall along the foreign genetic material to get into the plant system. Another method is that is the whisker mediated transformation. The gene of interested coated tangation whisker are mechanically inserted into the plant tissues to incorporate the gene. So another method is the liposome mediated transformation. Lipid vesicles are known as the liposomes, so which is carrying the gene of interest fused with the plant cell membrane along with the which allowing the incorporation of foreign material into cell. That is there. So another is the protoplast transformation. Protoplast transformation, the plants without their cell wall can be modified by using the transformation of this technique. Without cell wall, we call it the protoplast. So that is the directly the protoplast is also used to the gene transformation. Okay. Some examples of transgenic plants are the examples here. So there are the numerous examples means to produce the transgenic plants. So nowadays, these genetic engineering, this one is the widely used to produce the different plants. In the plants which are developed to the make a sustainable agriculture and the breeding technique also horticulture and agriculture techniques. These transfer means transgenic plants they are used. So first one is the BT cotton, that is the Bacillus thuringiensis toxin. BT toxin, nothing but BT toxin. The gene has been genetically incorporated into the genome of cotton plant to resist the pest infection, means bollworm infection, bollworm attacking The cotton mainly, which is caused by the bollworm only. So these Bt toxin to resistance to the or to stop the bollworm, okay, to growth and life cycle of bollworm. So that's one is agriculturally the Bt cotton is a tremendous changes to develop the to enhance the cotton production. Another one is the golden rice. So golden rice beta carotene is the precious of vitamin A. The BT means golden rice, which is rich in vitamin A only. That is the genetically incorporated the golden rice to eradicate the vitamin A deficiency in developing countries. So some developing countries, they are the children are that adults and children, they are suffering from the vitamin A deficiency due to the lack of the vitamin A when they are eat the food material or supplements. So and these genetic engineers, they are to develop the golden rice. So another one is the round ready soya beans. So glyphosate is chemically which is commonly present in the herbicides. To withstand glyphosate, round reed soybeans has been genetically modified. So means so these soybeans, they are to resistance to the glyphosate. Another one, the flavors are tomato. Flavors are tomato. So flavors are tomato or the genetically modified to suppress the ethylene, which are responsible for the ripening of fruit. This one is the most important nowadays. Is tomatoes is widely used. Another is the BT corn. So BT corn is also developed from the Bacillus thuringiensis taxon gene, has been genetically incorporated into the genome of corn. So these plants, which is resistant to the pest infection, nothing but bollworm infection. Another is example rainbow papaya. So rainbow papaya means ring spot spider disease in nightworm of papaya crop. So virus gene has been incorporated into the crop to resistance of this infection. Importance of transgenic plants. Another one is the some importance why we are to develop the transgenic plants. So the transgenic plants are of a great importance to make agriculture sustainable development. Here are some important transgenic plants, which is the pest and disease resistance. Pest and disease resistance. The crops has been genetically modified to make the resistance to the pest and insect infection, which can be crop with the withstand infection. So pest diseases. And another example, another importance is the herbicide tolerance. Herbicide tolerances means these herbicide can be harmful for the crop. So crops are genetically modified with to withstand herbicides. and increasing the yield capacity, enhance the yield capacity. To meet the global need for food crops has generally genetically modified to increase the yield crops. Nutritional increments in developing countries. The developing countries manufacturing manipulation, the major cases of death and diseases. So, to developing countries, malnutrition is the major cause of death and diseases. Nutritional enhancement can be done with the help of the transgenic plant. Hello? Uh, Another one is the abiotic risk tolerances to withstand the different types of abiotic risk crops can be genetically modified. Abiotic stresses also. So the improvement of shelf life, some crops can be easily to ripen. So to increase the ripening time, crops can be modified. So this is the uh, this one is the improving the life shelf life also. So now the conclusion here. Although transgenic plants have many advantages, their appropriate use requires careful consideration of ethical, environmental, and regulatory issues. Several factors that is the influence the transformation is the technique selection, including the kind of plants, the tissue, baby transfer, and the overall objectives of genetic alternation. The transgenic plants are ones whose genetic composition has been modified by the introduction of genes from other species. So this is the Here another diagram is shown to. So what is the okay? Now we discuss the plant proteomics and genomics. So what is proteomics here? The plant genomic and proteomics. Now we see here the meaning of proteomics. Of proteomics. So the term proteomics was coined in mid 1990s at the back draft of So a major challenge facing the plant biotechnology and other bioinformatics research community is the translation of complete gene DNA sequence data into protein structure and predicted function. Such a steps will provide key link between the genotype of an organism and its expressed phenotype. So this one is the main linkage between the genotype and so the growth of proteomics is the direct result of advances made in the large scale nucleotide sequencing of expressed sequence tags EST that is expressed sequence tags. Although mass spectrometry are more popularly MS technology means has been considered as a versatile tool for the examining the mm, simultaneous expression of more than 1,000 proteins and identification mapping of post-transformation modifications. So these methods performed in the latest array of technology resulted in the large-scale characterization of protein location, protein and protein interaction, and protein function. So mass spectroscopy, that is the mass spectrophotometer. So another in silico methodologies are being developed to identify protein interaction from genome sequence. For example, 6809 is the P2 protein interaction has been identified in Escherichia coli, and more than 45,000 has been identified in the East, and large number of these interactions functionally related. So types of proteomics. So structural proteomics, one of the main targets of proteomics investigation is to map the structure of proteome complex or the proteins present in a specific cellular organelles known as the cell map or structural proteins. So structural proteomics attempt to identify all the proteins within the protein complex and characterization all protein and protein interactions. Isolation of specific protein complex by purification can be simplify the protein analysis. Functional proteomics mainly include the isolation of protein complex or the use of protein ligands to isolate a specific type of proteins. It allows the selected group of proteins to be studied, its characteristic, which can be provide important information about protein signaling and disease mechanisms. So functional proteins, it mainly includes isolation of protein complex or the protein ligands isolated that the um, can be provide important information about the protein signaling and disease mechanism. Now, the significance of proteins, proteomics. Here, different types of the sequences are there. So, protein profiling bioinformatics has been widely employed in the protein profiling, where the question of protein structural information for the purpose of protein identification, characterization, and database is carried out. The spectrum of protein expressed in the cell type provide the cell with its unique identification. It explores how the protein complement changes in the cell type during the development of in response to environmental stress. Another one is the protein arrays. The protein microarrays facilitate the detection of protein interaction and protein expression profiling. The several protein microarrays example is indicate the protein arrays hold the grade to promise for the global analysis of protein and protein ligand interaction. The proteomics is to the phosphorylation. In the post-translation modification of protein, mass spectrophotometer can be used to identify the novel phosphorylation. So, measure changes in the phosphorylation. The state of protein takes place in the response to an effective and determining the phosphorylation sites in proteins. Identification of phosphorylation sites can be provided information about the mechanism of enzyme regulations and protein kinase and phosphatase involved. The proteomics approaches for this process has an advantage that can study all the phosphorylating protein in the cell at the same time. So proteome mining. So protein mining is the functional proteins approach. It is used to extract the information from the analysis of specific and subproteomics. In principle, it is based on the assumption. In principle, it is based on oil and assumption, all the like molecule selectively compete with the natural cellular ligand for binding site on the protein target. So coming to the genomics, what is the term of genomics was first introduced by the Thomas Frederick in 1986. It is the study of structure and functional entire gene of the living organism that we call as the genomics. The genome, which is refers to the basic set of chromosome. In a genome, each type of chromosome is represented by only once. Now, genomics is being developed as a subdisciplinary of genetics, which is developed to mapping the sequencing and functional analysis of genome. So, in the compound, what is the types of genomics? This discipline of genomics consists of two parts: are there structural genomics and functional genomics. So, structural genomics deals with the study of structure of entire genome of the living organism. There is the structure, the structural genomics. In other words, it deals with the genetic structure of each chromosome of the genome. It determines the size of genome of a species in megabases and also gene present in the entire genome of a species. So, functional genomics. Study of the function of all genes present in the entire genome is known as the functional genomics, which deals the with the transcriptome and proteome. The transcriptome refers to a complete set of RNAs transcript from the genome, and proteome refer to complete set of protein encoded by the genes. Classification of genomics. The genome can be classified as the plant genomics, animal genomics, eukaryote genomics, and prokaryote genomics. The plant genomics deals the study of structure and function of entire genes of plant species. That is the plant genomic. Another one is the animal genomics, deals with the study of structure and function entire genes of animals. Like that, the eukaryotic genomes only in the study of eukaryotes, and prokaryotes that deals with the study of structure and function of entire genomes of unicellular organisms like bacteria, that can be called as the prokaryote genomics. The role of genomics in crop plants. The role of genomics in the crop plants. The genome mapping is first completed in the free-living bacteria, that is Haemophilus influenza in 1995. So in plants, genome sequence was first completed in the Arabidopsis. So after that, rice, followed by the rice. Arabidopsis, mustard, and rice. So the new genome sequence work has been completed in more than 40 crop plants. List of some plants are there. That is the structure and functional genome. So genes to be mapped. The both genome research, both type of gene size, major genes, minor genes can be easily to map. The mapping of polygenic traits is possible by genome mapping, which is not possible by conventional gene mapping techniques such as recombination mapping and selection mapping. The genome mapping is done for the morphological productivity, resistance, quality, agronomic, and some special traits as discussed here. Morphological characters. It includes highly heritable traits such as shape, size, color of leaf, flower, calyx, and corolla. So these are the means the morphological character. Productivity states. Such characters differ from species to species, the productive depending on the different species. Resistance traits, such characters include resistance to diseases, insects or drought, soil salinity, soil alkalinity, soil acidic heat, and water logging in cold conditions. The quality traits, such as traits include nutritional quality, market the quality and keeping quality. So agronomic traits, such traits include earlierness, plant highest plant type. Another one is the special characters. Such characters include the genes controlling male sterility, self-incompatibility, photo and thermo sensitivity, toxic substances, apomixis, and another one is some adaptations. So gene mapping laboratories, another one discuss that one, and genome mapping in India. In India, the functional genome research project are looked after by the Department of Biotechnology and ICR means Indian Council of Agriculture Research. So the DBT means Department of Biotechnology instituted to work the several crops such as rice, wheat, maize, chickpea, banana, tomato, brassica, etc. The ICR has created genome mapping facility, the NG, NRCBP, IARI, New Delhi. These are there. So NRCPB means National Research Center for Plant Biotechnology and International Center for Genetic Engineering and Biotechnology. JNU means Jawaharlal Nehru University, New Delhi, and NBRI means National Botanical Research Institute, Lucknow. This is also improve that one. అయిపోయింది పక్క పెట్టిన అయిపోయిందండి ఇది పద పోదాం ఇక ఏమైంది ఎంత సేపు? నేను కూడా వస్తుంది పదండి.